you, Yelpi, for that introduction. And also, thank you all for joining this short talk. This will be a brief presentation on analyzing organoid single-cell data to study the various cell types that are involved in the development of alveolar organoids. But first, what are organoids? An organoid can be defined as a three-dimensional model derived from stem cells. These 3D models aim to study certain features of the organs from which the stem cells were obtained without considering the full complexity of the organ, but instead focusing on specific characteristics of it. For example, within an organoid, we can study aspects of development or regeneration, such as cell proliferation, differentiation, and communication. When discussing long organoids, there are multiple sources of stem cells, which results in different types of organoids. For instance, if we use basal stem cells, we can originate what are known as bronchosphere organoids, composed of basal stem cells, ciliated cells, and globed cells. Also, if we use alveolar progenitor type 2 cells, we can generate alveolar organoids, mainly composed of epithelial cells type 2 and type 1. As I already mentioned, the first thing that we need to generate these alveolar organoids are stem cells, but also sometimes are needed feeder cells, which are cells such as stromal cells or immune cells that help to stimulate the growth of the organoid and the formation of this three-dimensional structure. Also, feeder cells and epithelial cells are cultured together in a medium called matricial, which is a medium composed of extracellular matrix. And also, sometimes growth factors are added into the medium to promote the process of proliferation and differentiation. Growth factors and other molecules, such as signals, are important in the development of these alveolar organoids because they regulate the gene expression of the cells that are part of the organoid. This cellular signaling is a crucial and important part in the development because epithelial cells communicate with each other and with the other cell types, such as immune cells and stromal cells, within extracellular signals that activate intracellular signal pathways and at the end modulate the activity of some proteins called transcription factors. These transcription factors are proteins which bind into specific motifs in the DNA and ultimately affect positively or negatively the expression of the genes that they regulate. Also, an important concept to tell us about is a regulome, which is the group of all the genes that are regulated by a transcription factor. The dynamics of how these transcription factors regulate all the genes can be represented as graphs, and the characteristics of the different graphs may be influenced by depending on the data that we are using. For example, if we use single-cell technology, we can, in this case, single-cell RNA-seq technology, we can model the transcriptional activity within each cell type that that is part of the organoid. Essentially, this type of technology captures the transcriptomic of each cell type that form the organoids, in this case, a collection of organoids. Each cell is processed through a microfluidic system. Inside this microfluidic system, the cells are combined with chemical reagents that lyse the cell, release the messenger RNA, and then this messenger RNA is converted into complementary DNA through a retrotranscription reaction, is purified, amplified, and sequenced. After sequencing, we get a data matrix that we analyze to get insights of the different cell populations inside these organoids. And by using this type of data, we aim to answer the question of which are the specific regolons involved in the development of an organoid, an alveolar organoid. For this, we collect different metadata using public databases and select the experiments based on specific criteria. For example, in this table, I am showing you the experiments of alveolar organoids coming from mice experiments. And there are some characteristics that are important to say because we integrate all the experiments in one object, And there are some features that are important to consider. For example, the type of stem cell used for generating these alveolar organoids, the growing time at the moment of sequencing, the instrument and the protocol used for sequence. And after the data collection, we followed a modified CRAD pipeline to integrate the object and also to filter the cells based on the quality of the data. For this, the first thing that we did was use polydrops and double finders, which are packages that filter the cells based on adaptive thresholds, such as sequencing depth, mitochondrial percentage, total number of genes, protein coding fraction, and also remove the duplicated cells. After doing this, we normalized the expression values of the data using the single cell transform library, which is it's part of the CRAD pipeline and applies a negative binomial correlation analysis to normalize all the expression values. And these individual samples were integrated using the CRAD function called canonical correlation analysis that removes the batch effects at the moment of the integration. Following the integration, we can apply linear and nonlinear projection methods such as PCA and UMAP, and, for example, obtain this type of two-dimensional graph representations. This is the object that, well, this is a UMAP projection that shows us the different cell types, epithelial cell types that are part of the alveolar organoid. In this case, we have cells that are colored in different. These cells are group of cells are called clusters. Cells that are colored the same means that are cells that has a similar expression patterns. The interesting thing here is that, as we know, we have at least two cell types in lung, homostatic lung, AT2 cell type and AT1 cell type. But in this type of three-dimensional models, we are mimicking the process of regeneration. So we have transitional states coming from AT2 to AT1. But we analyze the different clusters that are part of the organoid, searching for the searching for the differential expressed genes. For this, we use the function called find all markers that find the top differential expressed genes between all the cell clusters. This is a heat map that shows these differential expressed genes. It ranges from purple to yellow, which purple means that there is no or low expression, and yellow means that there is high expression of the different differential expressed genes. What we can see here is that, for example, AT2, that is a cluster that has the high expression values of the canonical marker genes, such as lyse2 or surfactant proteins genes, have this intense yellow color at the top. But we also have these transitional states that lose the color of the expression of these markers and start to gain, the expression of other markers. We don't know too much about these transitional states. There are other articles that mention them, but there's not too much information of them. So we decided to label them as transitional. After labeled these cell types, we use a Python program called Scenic to infer the regulons that are characteristics of each of these cell types. This Scenic program uses a machine learning algorithm called GRNBoost, which gives us a co-expression matrix that gives us information of which are the different transcription factors and all the possible genes that are regulated by it. Also, this is just a co-expression matrix. There's another module that is part of Scenic program that is called RCStarget. This program searches for the specific motifs in downstream or upstream of all of these genes that are part of the regulon. And at the end, we have this matrix called AUCell that it's basically the sum of expression of all the genes that are part of each of these regulons. Now, this is the pipeline. If you want to take a photo, I have no problem. And also, When we get these regulons, we wanted to know which are the specific regulons for each cell type. We use the regulon specificity score, which is a matrix based on the Jaccard index that tell us how specific is a regulon for each of these cell types. This regulon specificity score ranges from 0 to 1, which 0 means that the regulon is shared between all the clusters, and 1 means that the regulon is completely specific for one cluster. We later make enrichment analysis of all the genes that are part of a regulon and search for biological process. For this, we use gProfiler, which is an online tool that gives us the results that there are some regulons that are related with developmental process, collagen biosynthesis process, and cellular responses to organic substances. This is really cool because these regulons are genes that are related with a specific transcription factor, but there are no studies that binds these regulons to the process that I already mentioned. So now, as a keynote, we already have this the integration of all the organoids data sets of alveolar organoids. We characterize the epithelial cells and reveal that there are different populations involved in the alveolar organoid. We search for specific regulons inside these cell populations, and also we found that there are regulons in transitional populations that enrich to the process of development. But this is the results of the enrichment analysis. We also want to know which are the transduction pathways of how these regulons are activated for which kind of signal. So also I want thanks to CONACYT, which is the institution that is financially supporting my studies, and LABIC members, which is the laboratory where I'm working on. And thank you very much. I don't know if you have questions.
Thank you, very interesting. Out of curiosity, how easy is to do single-cell ataxic on organoids?
Ataxic?
And are you thinking of
Yeah.
it?
I do not analyze ataxic, but there are some papers that use the ataxic and also single cell and analyze.
Yeah, but how easy do you think it is to do it on organoids?
No. I don't think that it's easy at all, because at least we are studying epithelial cells, and there are results that shows that there is not a lot of heterogeneity inside these cell types of these populations. Yeah.